All right. Is the record? Yes, the recording is on. Okay. So the title of my talk is an algorithmic barrier to neural circuit understanding, and the video that you see on the title slide is that of the Hydra, right? And the Hydra has what the biologists call a nerve net, which is just a network of neurons which control movement, and you can see this. And there are techniques now to image activity of all of these neurons, and basically the neurons light up when they are active, right? And we'll see more of this as we go along. But before starting off, as a sort of fun thing, I wanted to sort of get back to this whole interface of neuroscience and computer science. And many of you might imagine that neuroscience and computer science intersect with artificial intelligence, and indeed early efforts at AI look to the brain as proof of principle. But in fact, I wanted to point out that the relationship is actually much older and possibly deeper. So this is from a report on the EDVAC by John von Neumann, and EDVAC was an early stored program computer, and in fact the first exemplar of what we now call the von Neumann architecture, right? And so this is the architecture that underlies by and large all digital computers today. And von Neumann was very interested in neuroscience and of his time. And in fact, the report itself has section on neuron synapses, and he talks about excitatory, inhibitory types, and in fact even writes in fair amount of detail on it, right? And so certainly von Neumann was very influenced by neuroscience, and presumably this also played a role in the design of the architecture itself. So it actually goes back much more than simply being another proof of principle for AI, right? So be that as it may. So let me just sort of give you a 2 or 3 slide neuroscience background, right? And so here's sort of a picture of the brain, of in fact the human brain, and so primarily, in addition to sort of structural things such as blood vessels and other kinds of cells, the 2 main cells of interest are what are called neurons and glial cells. All right, neurons are currently thought to be the main players in computation, right? But increasingly, the role of glial cells, which had sort of earlier been thought of primarily as having a structural role, now increasingly there is work that shows that they also sort of modulate neural activity, and neural activity modulates the activity of glia and so on. And in addition, there are estimated to be about 10 times as many glial cells as neurons, right? So our sort of understanding of their role is still evolving as well. And there's a slew of recent very interesting work that shows in fact glial cells being responsible for very specific aspects of behavior, but which I won't get into today. So the human brain, for example, is estimated to have the order of 100 billion neurons, and estimated to have of the order of 100 trillion connections, which are called synapses, right? And so here is sort of a very simple schematic cartoon view of what is called a spiking neuron, which is thought to be the primary type of neuron in mammals, right? And so this sort of green cartoon is a schematic of a neuron, and typically neurons have these processes or branch processes called dendrites, which form synapses with other neurons. So this is and then the neuron itself sends its own output via a process called an axon, right? So typically another neuron's axon will connect with this neuron's dendrite at this junction, which is called a synapse, right? And neurons communicate with each other using these stereotypic pulses, which are called action potentials or spikes. All right, and here all that matters. So the shapes of these pulses are all the same. All that matters is the relative timing of the pulses. Right? And every neuron has an internal variable called its membrane potential, which is the potential difference between the inside and the outside of the neuron. Right? And this potential difference tends to be at a stable equilibrium called the resting potential. Right? And when these action potentials or spikes come from other neurons, that tends to move this membrane potential, about its resting potential. So here the resting potential is sort of taken as 0, but when this spike comes in, for example, that kicks it up, and every so often the membrane potential hits a certain threshold, and when it does, this neuron fires its own spike, which is then communicated to the neurons that it is connected to. Right? So this is sort of, again, a very necessarily a description that is skipping a whole lot of details, right? And a lot of which we understand very well now in neuroscience. But this should give you a sense of what a neuron is like, right? And of course, all of these, this whole, the membrane potential and so on, and its dynamics are governed by things like ion concentrations between differentials between the inside and the outside of the cell, things like ion channels and other details which I'm sort of skipping now. Right? And oops, do I see a question?
Yeah, sorry.
Yeah, Tivraj,
in your video that you are showing at present, the animation, it shows that there's some kind of time delay in this information flow. Is this correct?
Yes. Yes, that is correct. So so neurons typically time delay in the sense of, yes. So these spikes come into the neuron, they cause these changes in membrane potential, and that triggers action potentials in this neuron. But this happens at the time scale of milliseconds, right?
Oh, okay.
So all right, so it is, yes, there is a time delay, but it is much smaller in comparison to the time scale of behavior, which occurs in the time scale of seconds.
Thanks.
Right?
Thanks.
Yeah. Okay. So again, this is I'm skipping a lot of details, so I should emphasize that. So there are a couple of other notions which are absolute refractory period and exponential decay of the effect of a spike on the membrane potential, but these I'm going to skip now in the interest of time. One other quick point I wanted to make is that in contemporary AI systems, things like artificial neural networks, we have these real valued variables, and which are typically taken to be abstractions of spiking rates of these neurons, right? And so again, this goes back historically, and still in neuroscience, when people do experiments, one often is able to find specific types of neurons which whose firing characteristics can be correlated with certain aspects of the stimulus or of behavior, right? And so and therefore with artificial neural networks, the firing rate is abstracted as a real variable, which is then used in these networks. But now there is also the importance of precise spike timing, right? So what this abstraction that I just told you about sort of loses is the is information about the timing of individual spikes, right? And this has been observed to be important in certain modalities such as audition, olfaction, and tactile perception, right, where one can find aspects of stimulus or behavior that are correlated with aspects of spike timing. Okay, and then there are learning rules. So there's a learning rule called spike timing dependent plasticity, which strengthens or weakens the synapses depending on the precise, arrival time of spikes versus the time of firing of spikes, right? And then, oops, what did I do? Yeah, and then there's also other evidence, namely with fast processing in the visual system. So, so what this is saying is that given the sort of gross anatomy of things like the visual system and how fast animals can respond to visual stimuli, that only allows us just simple calculations suggest that only allows time for a few spikes to have been fired in that duration. So there isn't a whole lot of time to measure spike rates and so on. So in a way, the nervous system in those cases, would have had to use the spike timing in order to perform computation. But this is again another aspect that I thought many may not be familiar with. So I thought I'll put this in. So and another aspect of neuroscience, over the past couple of decades, there has in fact been extraordinary progress in experimental techniques in neuroscience. Right? And I'm going to take the next 10 or so minutes to walk you through some of these. All right? And so the first one is what is called connectomics. All right? So connectomics is so what is connectomics? So the connectome of an organism is the exact structure of the networks of neurons in its nervous system. So the underlying graph of the brain, if you will. And now, of course, because neurons are really small and they are packed so tightly, determining this is quite difficult, right? So and further complicating things is the fact that synapses, which are these junctions between neurons, are actually significantly small, of the order of about a few nanometers, right? And therefore, as a result, they cannot be resolved by light microscopy. They cannot always be resolved by light microscopy, depending on the synapse. So, so really, the only foolproof way of telling whether an axon and dendrite have formed a synapse is to use what is called electron microscopy, which has sort of resolution at the level of single-digit nanometers. Right. So so this the broad technique is fairly old, and the first connectome that was determined was that of this roundworm called C. elegans, which has 302 neurons, and this was determined in 1986 by Sidney Brenner and colleagues after a 15-year effort. Right, and all of this was manual. Right, so they literally in Cambridge had this sectioned C. elegans worm, and printed out this thing and colored it with sketch pens, and because it was so huge, it took a lot of time. And the only other complete connectome was, I think, in 2016, which is this other tadpole larval connectome of the sea squirt, which in fact had about half the number of neurons. But since the early 2000s, there has been a sort of move to automate these techniques, given advances in computer technology. Right? So whereas the folks in the 1980s were doing this sort of manually, now the idea was to sort of automate the acquisition of these electron micrographs, and indeed try to use computer vision to automate the reconstruction as well. Okay, and so let me sort of walk you through a video of how connectomics is done these days. So typically you have brain tissue that's prepared by staining it with heavy metals, because you're using electron microscopy. So you want to see different components of your cell differently, and this is embedded in this hard resin, and that is sliced with a diamond knife. Just 1 second. Right. So this is going to be a video, and what you'll see in the video is this person who has a dish which has these slices of brain tissue from a mouse, right? And the video will basically fly through it, right? And so you'll see what I mean, right? So these small boxes are pieces of tissue from the mouse cortex, in fact, right? And as you fly through it, and what you see are electron micrographs at various resolutions, and now you're beginning to see the cell bodies, right? And so in your computer, you can sort of put this entire stack of electron micrographs together, one on top of each other, and just as you would color with a sketch pen, you let your computer do the coloring, right? So in this case, they are tracing this neuron which they colored red across the entire stack of electron micrographs. And when you do this soon enough, you find that the entire neuron sort of magically emerges out of your electron micrographs, right? And so here is a neuron colored red and another neuron colored green, right? And then you can in fact go into an appropriate electron micrograph and zoom in onto the synapse. And electron microscopy offers great resolution, so you can in fact even see the synaptic vesicles that hold neurotransmitter. And so so when you reconstruct all of the neurons in this in this entire volume, you actually find that the neurons are really squished together, right? And there is really very little space in the brain, right? And everything is just squished together, and yet remarkably it manages to work and work rather well, right? So this is always something that I find fascinating. Anyhow, so yeah.
Okay, so the one that you showed, that cube kind of structure, those are all neurons, is it? I mean,
Yes. So each, oops, let's see. Maybe I can skip through. So
okay.
so each thing so each thing is a different neuron. Yeah.
Oh, okay. So this layered structure that we see,
yeah,
and people keep saying that deep networks are somewhat inspired from brain. It's not really true, isn't it? Then?
Well, so the cortex, obviously, for example, is sort of thought to have some sort of a layered structure, but there are feedback connections as well. So there are
that layered structure I'm not able to see here.
Right. So this is in fact okay. So what I should say is this is a fairly small piece of tissue. So if I recall right, this must be 1 micron cube or smaller. And the best that people have been able to do so far is a millimeter cubed, and that is very recent as well, right? And for example, this is from the fruit fly, right? So fruit fly is this. So if you leave a ripe banana out in your kitchen and you come back in the morning, you find these really tiny flies. So those are fruit flies, right? So those are called Drosophila melanogaster. And so for the fruit flies, they are close to finishing the reconstruction of the entire connectome. And fruit flies have of the order of 100,000 neurons, all right? And however, a mouse has of the order of 75 million neurons, right? So it's a significantly large number. And here is, for example, a tweet by Jeff Dean, right? And this was, I believe, last year, right? So they wrote this sort of position piece where they want to be able to provide a complete mouse brain connectome that includes all synaptic connections. Okay, so now you may be a bit confused because Jeff Dean is from Google, as you know. So what is Google doing here? All right. So again, to take you back a little bit, in the first decade of the 2000s, while a lot of the electron microscopy, etc., was automated reliably, the big roadblock to getting connectomes was the 3D reconstruction, right? So the computer vision algorithms weren't as good, but a few years ago, with a lot of generous help from Google, there are deep learning techniques that can now do the reconstruction of the 3D structure quite well, right? And so that has really helped in in improving how this is done. And now you might say, well, why would Google care about this? And so the reason Google cares about this is that they want to use these reconstructed structures to seed their own deep networks to see if they will do well, and they have a lot of money in the bank to be able to do this sort of thing. So that's what they're doing. And there was so people were speculating as to how much a mouse connectome would cost, and many think it would cost of the order of $1 billion, right? So this is sort of serious work that's been going on. So connectome is one thing. The other is both in terms of inferring neural activity of neurons, as well as the ability to stimulate or silence them. In other words, ability to perturb them. All right. And here again, the old technique, and by old technique I mean the predominant way, the predominant 20th century way to do this was via electrodes, right? And people still use electrodes because they offer certain advantages that these new techniques don't yet have, right? And the idea is that you would, because neurons are inherently electrical entities, that you can have an electrode and you can record activity of either a single neuron intracellularly or a bunch of neurons extracellularly. And but again, the problem here is that even if you have a large array of electrodes, typically the best multi-electrode arrays can get you a few hundred, maybe 1,000 neurons, right? And even a mouse has of the order of 75 million neurons. Right. Likewise, the way to stimulate neurons would be to inject current via your stimulating electrode. So this was the old, now old way of doing it, which is to say that people still do it because it offers advantages. But more recently, there have been a slew of new techniques. So one is called calcium imaging, and this is the idea that when a neuron fires briskly, all right, there is calcium released inside of the neuron in order to, which is important for the process of firing. So they have, so what they have is they have sensors for the calcium, which will fluorescent sensors, such that when you then probe the neuron with lights of certain wavelengths, it will reflect light back with certain wavelengths. So you know that then at that point that neuron has fired briskly. And so that is called calcium imaging, or variants of it are called 2-photon calcium imaging and so on. Then there is what is called optogenetics, right? So optogenetics is the ability to stimulate or silence neurons via light. Now you might say, how is that even possible? So what they've found is they have adapted ion channels from organisms such as algae, such that when light or specific wavelengths you shine, so first of all you express these engineered ion channels in your neurons, and then when you shine light or specific wavelengths, those ion channels open up and let in ions of specific types, which causes the neuron to be stimulated. Right, so what you can do is with a laser, you can pick which neurons you want to stimulate and just shine light on them, and they will get stimulated. And there are, with other kinds of ion channels, you can now also silence neurons using similar ideas. There's also ways of doing this via temperature, and I'm going to quickly walk you through a couple of papers on it. And not only can you do these things, because light is sort of easier to deal with in these ways than electrodes, you can do this at ever larger scales and in awake behaving animals. So let me just quickly run you through a few examples. So here's a paper that was out about 4 years ago now, and this is in the fruit fly, right? So Drosophila. And what they did was they expressed these what are called trip channels. So trip channels are channels that open up not due to light but due to temperature. So if you raise the temperature from 25 degrees centigrade to 31, these channels will open up, and the neurons in which these channels are expressed will then fire, right? So again, through genetics magic that is available, genetic toolkits called GAL4 lines, which are available in Drosophila, and I won't get into this in more detail than that. So there are of the order of 2,000 different GAL4 lines. So each GAL4 line is one in which a different subset of neurons in Drosophila expresses these channels, right? So what they did, this was a large scale, so they inconvenienced 400,000 flies, across 2,000-ish GAL4 lines. And what they did was they constructed this huge GAL4 line to behavior probabilistic table. So what they asked is, well, if I stimulate this subset of neurons, what does the fly do, right? So it may walk away, it may try to fly, it may try to mate, etc. So, and then they, again, using a lot of computer vision, both to characterize behavior and a lot of these techniques, they are able to build this big data set, right? So this is something people are doing. Now, a few years before this, they did something similar with optogenetics, but with larval Drosophila. And here again, they had something like 37,000 plus animals, right? And similarly, so here is a paper that looks at imaging the mouse cortex. So, so what they did here was they took out the back of the mouse skull, right, and they implanted this glass crystal skull, right, and then this allows them to image activity in the mouse cortex. So let me tell you what you're seeing here. So the thick tree-like structures are blood vessels, all right, and the white dots that you see are individual neurons in the mouse cortex that are firing briskly, right? And this is an awake behaving mouse, right? And so this sort of imaging people can do. And in this video, the estimate is that you can see of the order of thousands of neurons whose activity you can infer, right? Okay, let me just check that I'm still on. Okay, I am. I think I am. Okay. All right. So not just can people image subsets of neurons. In some organisms, you can in fact do image activity of every neuron in the brain, or almost every neuron in the brain. So here is a larval zebrafish. Right. So zebrafish is a species of fish, in fact, native to the Indian Ganges. Right. And the nice thing about the larval zebrafish is that at the larval stage, the head is transparent, so you can see the brain from the outside, right? And what this allows people to do is to do calcium imaging in the live larval zebrafish, right? And this is what you're seeing. And what happens is that they sort of embed this larval zebrafish in this agarose gel, where it cannot move, so they can image, and or it may be able to move only small parts of its this thing, and then they show it stimuli and so on, and record activity, right? And this is what is called whole brain imaging. And the tiny dots of light you see are individual neurons that are lighting up, right? Similarly, people can do likewise with C. elegans, which was the roundworm for which the connectome is known as well. So here is a C. elegans that is sort of crawling around in this dish, right? And you can in fact image calcium activity as it's moving along. So you can track it here, and you can sort of see which neurons are firing away, right? And this is, of course, the hydra, right? Which I had on my title slide. Similarly, so each of the neurons that are lighting up are firing away when this is happening, right? And this is likewise with the fruit fly, the adult fruit fly. And here again, so this fruit fly is walking on the styrofoam ball, right? And on the left, you see the brain that is being imaged, all right? And In the top movie, it is being given a puff of ethanol, right? And when it's being given a puff of ethanol, you see this the square light up, right? So, and when it receives ethanol, a lot of neurons light up, right? And so it's just a demonstration, right? And likewise, the bottom movie is similarly, but with flashes of UV light, which fruit flies can perceive, right? And when UV light is flashed, this square box is shown just for illustration. All right. Okay. So good. So hopefully you have a sense of what modern neuroscience can do, and in fact you can apply many of these techniques together. So for example, you can both image activity as well as perturb activity of neurons. So here's the paper that shows the ability to optogenetically stimulate arbitrary subsets of neurons during whole brain imaging, right? In the larval zebrafish, right? And this is again about a couple of years old now, right? So these are things that people are routinely beginning to be able to do. Right? So and indeed, there are several national and international initiatives that are aimed at building technologies to accelerate techniques such as these. Right? And so there's the US Brain Initiative and the Japanese Brain Minds Project, and projects in China, Korea, and Australia, to name a few. So sort of the goal of many of these projects is to give us techniques that will enable us to understand precisely how mechanistic computation by the network causes behaviors in animals. Right? And so therefore we are sort of at the cusp of an era of large-scale neuroscience at the network level. And what we want to know is precisely how is mechanistic computation by the network causing the behavior in question. Right? And we seek a sort of comprehensive empirical understanding, right? And one thing I should note is that when I say causing, in other words, we are not necessarily satisfied by just a correlative account, which is to say that we want to know more than knowing, well, which neurons activity is correlated with which aspects of stimulus or aspects of behavior. We want to understand the exact mechanism. Right. And so to understand mechanistic computation, we will require efficient algorithms. Well, why is that? Because of just the scale of nervous systems, right? So even a mouse or even your Drosophila has of the order of 100,000 neurons. So no excuse me. No grad student or postdoc can be expected to sort of keep track of individual neurons from these 100,000 neurons and figure out how exactly each behavior happens. It will simply be too slow. All right. Right. So the algorithms will basically seek to prescribe the smallest number of experiments necessary that scale as a function of the number of neurons in the network towards this goal. And establishing this type of causal understanding will require multiple perturbational experiments, right? So perturbational could be, for example, optogenetic that I talked about. And now it's unclear exactly how many experiments you need, right? And an experiment could entail perturbing activity of a subset of neurons while imaging their activity and attempting to elicit behavior. And the specifics of the next experiment prescribed by the algorithm could depend on the outcome of the current experiment. So fairly generic sort of experimental setup to try to understand these nervous systems. So we are treating the problem of experiment design in neuroscience as an algorithmic problem, right? And invoking tools from computer science. And of course, since all of us here are from computer science, we know that there are problems with fast algorithms and problems with not so fast algorithms, right? And so the question is, which class of problems does those corresponding to neural circuit understanding fall in? And this will be the question. And in other words, how efficient can the most efficient algorithms be, right? Okay, so let me just give you a sense of what I'll be showing, if briefly, before I get into the details. So one, I'll basically prove some NP hardness results, basically. But furthermore, so what those NP hardness results imply is that establishing the most extensive notions of empirical understanding, need exponentially many experiments in general, unless P equal to NP, right? But what also turns out to be the case, which I'll get to, is that the feasible experimental regime is in fact one where you can only do less than linear number of experiments in the number of neurons, right? And again, I'll get to this in a few slides. And so, so this sort of brings us to back to the drawing board in a sense of trying to be forced to rethink what would understanding nervous systems mean, and which notions of understanding can we tractably hope to accomplish, right? Okay, but again, more on this later. So let me quickly walk you through the experimental scenario. So you have an individual animal doing a certain behavioral task, and you want to obtain a comprehensive understanding of how its neural circuits cause the behavior to be manifested at the present point in time, corresponding to a single state. And I want a causal understanding, not just a correlational one. And then nervous systems also have behavior, right? So I need a notion of a behavioral readout, right? Now behavior is a complicated thing, right? So now I might wish to pick up my water bottle, right? And I might do this. And now you might go in and you might silence a few neurons in my brain and ask me to do it again. Now I'm trying to find my bottle, and I sort of find it, and but ever so slowly I'm able to pick up the bottle. Now whether I accomplish the behavior correctly or not depends on your definition of what correctly executing the behavior entails, right? So that has to be provided by the experimenter, right? Which is saying that which kinds of behaviors are said to be correct and which are incorrect, right? So given such a partition, and here is you can think of a rodent with an odor discrimination task, etc., which I won't get into. So then the question is, what do you mean by understand? Right? What is understanding? Right? Which is again a very loaded question, and it's an especially loaded question in biology and in neuroscience. Neuroscience because of biology, because biology is messy, right? So biology has details at several levels of description, right from the level of gross anatomy, right, down to circuits of neurons, down to individual molecular aspects of neurons, and what sort of neurotransmitters, what sort of ion channels, and even at the level of genes, which genes influence which kinds of behaviors, and so on. Now, depending on the particular neuroscientist you ask, they may be interested in specific aspects of it. So if I come in and say that, look, I'm only interested in the circuit level, this molecular neuroscience person may come in and say, hey, that is quite inadequate because you are missing out on these other aspects that are important. So, oops, what did I do? Yeah. So, so there's no standard definition, for what exactly constitutes understanding in neural circuits. And there exist multiple concomitant descriptions that might, for example, include details spanning different spatial and temporal scales. And this is a sort of problem with biology. In fact, there's also a problem with computer science, but we don't think about it because, we use abstraction, right? So we never think about, when we're writing code, which transistor in our microprocessor will will execute, which set of transistors will execute this instruction, right? Because it doesn't matter. Whereas in biology, because we don't know what to look for, everything could be potentially important. Okay. All right. So we want to have our analysis apply broadly to many such reasonable notions of understanding, especially those that span the circuit level. And now, again, what is understanding, right? So understanding is the ability to answer questions about the subject of understanding in short order, fairly fast, right? Without taking unduly large amounts of time. Excuse me. And so, so if I claim to understand something very well, right? Let's say comprehensive notions of understanding, then I should be able to answer certain basic questions about computation at the circuit, in the circuit leading up to behavior. So what I'm going to do here is to posit a list of such basic questions that involve determining certain subsets of neurons that causally participate in computations, leading to the said behavioral readout. And again, the idea is not to sort of specify an exhaustive list, but merely to demonstrate the existence of multiple questions that cannot always be tractably answered. Okay, so this will be easy. Oops. Yeah, this will be easy for the computer science audience. So, so again, here, so sort of a more general setting would be that I have this individual organism whose certain behaviors I wish to understand. So I'm doing a number of experiments, and maybe what I'm doing is I'm collecting a bunch of data, right? So one question that the experimentalist might be interested in is, well, what data should I collect? And then, in order to obtain my understanding, I then work on that data to answer questions, right? So that is one way of setting it up. So I'm going to use notions of reductions in theoretical computer science, which all of you are familiar with. So I'm going to do this fast. So reduction is just that if you have two sort of abstract problems, A and B, all right, a reduction is a quick recipe that, given an instance of problem A, gives you the ability to construct an instance of problem B, right? Such that you also have a mapping back from the solution of problem B back to the solution of problem A. So typically, these will be decision problems. So yes instances of B will correspond to yes instances of A, and vice versa. But that doesn't have to be. So you can have another function that maps back. All right. So what I'm going to say is that I'm going to sort of claim or that any notion of comprehensive understanding ought to enable me to solve a certain question about the neural circuit, which I'll call problem X. Right? So I'm going to list a bunch of problems and say that, look, if you claim to comprehensively understand the circuit, surely you ought to be able to answer this question about the neural circuit form. All right. So so formulating multiple questions about mechanistic computation. And again, this goes back to sort of colloquial notions of neural circuits being necessary and sufficient for the expression of fixed behavior. And these are sort of, again, notions that people talk about in neuroscience, all right? Although it is almost never defined rigorously. What does it mean for a circuit or for a subset of neurons to be necessary? And what does it mean for a subset to be sufficient? All right? But people use these terms, right, in sort of colloquial ways, if I may say so. So so yeah, so I already said this. So any comprehensive notion of understanding ought to enable us to answer one or more of these questions in short order. So I'm going to now walk you through some quick definitions before we get to the results. So a subset N of neurons is said to constitute a degenerate circuit for a behavior B if B can be successfully elicited with the silencing of all neurons except those in N, right? So I'm going to silence all the neurons except the neurons in the subset N, and if upon doing so the individual animal is still able to express the behavior, I say that, look, this subset of neurons is a degenerate circuit. It's sort of sufficient to express this behavior, right? And now there always exists at least one degenerate circuit, right, which is the entire network. All right. Although in practice there exists many degenerate circuits, and in fact we have a lot of circumstantial evidence for this. So, for example, by some estimates, thousands of neurons in your brain and my brain die every day, yet we hardly seem to notice, right? So, so that is sort of one this thing, and and again in animal experiments as well, similar things have been observed. Now, presence of neuron in a degenerate circuit does not necessarily mean that it's causal, right? Because there could be another degenerate circuit identical to the present one, except for the absence of the neuron in question, right? So we define another notion of a minimal degenerate circuit, which is a degenerate circuit with the additional property that no proper subset of that set N also remains a degenerate circuit for B, right? So you sort of, in a way, you can imagine that you start from the entire set of neurons, then you shrink your set of neurons smaller and smaller until you reach a subset such that if you try to shrink it any which way, that the behavior is no longer correctly expressed, right? And that is called a minimal degenerate circuit. Now there could exist multiple minimal degenerate circuits, right? So depending on how you sort of start whittling down your subsets, you could arrive at different minimal degenerate circuits. So it may not necessarily be unique. However, in the context of a particular minimal degenerate circuit, each neuron in it is necessary to elicit the behavior, since silencing it abolishes the behavior. Okay, and you can also talk about a minimum degenerate circuit, which is a minimal degenerate circuit of the smallest size. So next we sort of switch gears to ask, well, what is a small set of neurons that I could silence that will result in the behavior not being expressed, right? So here is a definition. So I call this a vital set. A subset N of neurons constitutes a vital set of neurons for a behavior B if N has a non-empty intersection with every degenerate set for B, right? So you can imagine that suppose you knew all the degenerate sets for B, right? And you took the intersection of all of them and you found that there is a certain subset of neurons that occurs in every degenerate set, right? Now, if you went in and silenced those subset of neurons, then it cannot be that the behavior can still be expressed, because with those neurons silenced, no degenerate set can be active, all right? So a vital set, furthermore, of size k is called a k vital set. And you can also talk about a minimal vital set, which is a vital set with the property that no proper subset of it is a vital set as well. All right, so this I already said. So what's the problem setting? Well, suppose I gave you the simulation of the entire neural circuit starting from an initial state. Answer questions pertaining to finding sets of the aforementioned type, right? And here are the 6 questions that that I have, that I will show are basically don't have general sub-exponential algorithms unless p equal to np. So first question is, determine a degenerate circuit of size k, or indicate if none exists of that size. Determine a minimal degenerate circuit of size k, or indicate if none exists of that size. Determine a minimum degenerate circuit. Determine the set of 1 vital sets, or indicate if there are no 1 vital sets. Determine a minimal k vital set, or indicate if none exists of that size. And how many minimal k vital sets exist, right? Okay, so now you might say, well, sure, you're going to show some NP hardness results, and but how does that have a bearing on the number of experiments that experimental neuroscientists can run, right? And this is so what you can do is you can sort of imagine that a typical experiment where you're imaging neural activity and stimulating neurons is of the following type. So let's say you already know the connectome, all right? So again, this is sort of assuming the sort of best ideal case, some of which we cannot yet do, but be that as it may. So so suppose you can infer activity of all of the neurons, and you can specify which neurons to stimulate and silence. All right. So this is one experiment. All right. So this one experiment I can simulate with linear number of steps in my algorithm. Right. So what this is saying is that suppose there existed a sub-exponential schedule of such experiments that could get me the answer, then I would also have a sub-exponential algorithm, which is just off by a factor of n from the number of experiments, right? And therefore, which would imply p equal to np, of course. So this also has implications on the number of experiments, right? Okay, so what's the problem like? Well, so the first problem is, given a whole brain behavior and a positive integer k, does there exist a degenerate circuit for B of size k? All right, again, this so the claim is that, if you comprehensively understand the circuit, you ought to be able to tell if there exists a degenerate circuit of size k, right? Okay, so all of you know about NP completeness, so I'm going to not say anything about it, right? So I'm going to reduce the clique problem to this problem, right? And recall that a clique of an undirected graph is a subgraph of it, such that every pair of vertices in the subgraph has an edge between them. So here is a clique of size 5, right? So within these 5 vertices, every pair is connected. Here is a set of 4 vertices that don't form a 4 clique because there's no edge between these two, right? So the clique problem is a decision problem, which, given an undirected graph G and a positive integer k, asks if G has a clique of size k, right? And this is known to be NP complete, right? So I'm going to do a polynomial time reduction from clique to DC, which is the degenerate circuit problem, and here's how that will go. So I'm going to take this undirected graph. I'm going to put in a single neuron for each vertex, and I'm going to put in bidirectional connections for each edge between these neurons. Right? I'm going to throw in what I'll call a sensory neuron and a motor neuron in quotes. Right? So and these I'll call interneurons, and so the sensory neuron will be connected to every interneuron, and each interneuron is connected to the motor neuron. Right? So this neural circuit will have the following properties. So the behavior is evoked. So behavior, so the motor neuron firing signals the performance of the behavior. So behavior is evoked when the entire network operates, right? So right, and the way this will work is that when the sensory neuron fires a single spike, ultimately the motor neuron will fire a single spike, right? And that will cause the behavior to be executed. So it's a very simple circuit, right? So so one, behavior is evoked when the entire network operates, but furthermore, what the construction will ensure is that there exists a k+2 size degenerate circuit in the above network if and only if the undirected graph has a k clique, right? So and this is how that goes. So you have bidirectional edges between interneurons. You have a sensory and a motor neuron. The stimulus elicits a single spike from the sensory neuron, as I just said, and single motor neuron spike causes behavior. So the conduction delays are the same for each pair of the following neuron types. So the sensory to inter have the exact same conduction delay. The inter to inter also has the same conduction delay, and the inter to motor has the same conduction delay. Right? And so the spike from a sensory neuron elicits a single spike in each interneuron. So the synapses between the sensory to interneuron are very strong, and therefore a single sensory neuron spike will cause us each of the interneurons to fire a spike. right? So if all interneurons spike concurrently, the motor neuron spikes, right? So this is sort of when the whole circuit is intact. Now, if there is a pair of volleys of spikes from a set of k interneurons, the motor neuron also just about spikes, right? So in other words, if there are k interneurons which send one set of spikes, and at the next time instant they send another volley of k spikes, then that is sufficient stimulation for the motor neuron to spike, right? So you can set up this motor neuron for this to happen. So spiking from a set of less than k interneurons, however, can in no event cause the motor neuron to spike. So, oops. Yeah. And finally, if an interneuron is not receiving input from a sensory neuron, it needs concurrent spiking from at least k minus 1 other interneurons to produce a spike, right? So already you can see that I'm setting it up so that these interneurons can send a volley of a k-sized volley of 2 spikes to the motor neuron if and only if the underlying graph has a clique, has a k clique. Okay. So I think I'm going to skip these. So this is basically addressing some questions about, well, in biology, it's not necessarily the case that connections between neurons are bidirectional, but you can sort of embed this in a larger graph where you have an arbitrarily small number of such pairs of neurons. Right? And so I'm going to skip this. Right? And so what I've shown is a reduction from clique to DC. And then a sort of claim that any notion of comprehensive understanding ought to enable us to answer the degenerate circuit question. All right. So there are 5 more reductions which I'm going to skip today. All right. But now for the caveats. All right. So now, of course, all of you know about NP hardness, and NP hardness is a worst case result. Right. In fact, there are several NP hard problems which we routinely solve many instances of without a problem, right? So you may say, you may roll your eyes and say, well, so what? So in other words, what does this worst case result mean? It says that there exists a subclass of neural circuits which probably require exponentially many experiments unless p equal to np. Right? But this is, so on the one hand, this is a rather simple class of circuits, right? So if you recall my reduction a couple of slides ago, it wasn't really doing any sort of complicated computation. Right? It was just taking your input and sort of relaying it to your motor neuron. Right? And so on the one hand, you might expect that circuits doing more sophisticated computations might be at least as hard to understand comprehensively. All right, and also analysis for a single state and single behavioral readout, right? But typically, you might want to understand multiple behaviors from a single individual and how its neural circuits work and so on. And the other aspect is that this is only saying, well, is this neuron important or not? But it may only be important towards the initial half of the behavior, not the latter half, and so on. So at what time is this neuron important is not being addressed in this question. All right, and this is this I'll skip, not an impossibility result. But before that, so sure, you have NP hardness results, you could still have fairly efficient algorithms. But then how many experiments can you actually do is the question. All right, in your individual animal. Right? So to obtain a rough upper bound, so I'm going to consider how many experiments might one hope to perform during the lifetime of an individual animal and how this compares to the number of neurons in it. Right? So what I'm going to assume is that each experiment that we have, which deals with behavior, takes a second, which corresponds to the behavioral time scale, and that there is no gap between successive experiments. So I'm going to run experiments back to back from cradle to death, right? And these are again very conservative assumptions, right? So clearly you cannot, it is unclear how you could do better than that. Right? So what I did is plotted estimates of the average lifetime of a number of organisms in seconds, which also serves as a proxy for the number of experiments one could perform in an average lifetime as a function of the estimated number of neurons in their central nervous system. Right? So in other words, if an animal has n neurons, what function of n number of experiments could you do in the animal's lifetime? Right? And so here is a log plot of estimates of the average lifespan as a function of estimated number of neurons in the nervous system for C. elegans, fruit fly, mouse, octopus, marmoset, human, chimpanzee, rhesus monkey, and elephant. Right? So those are the blue dots. And these are respectively log of x. This is x squared, x cubed, and 2 to the x. Right? And there's a log plot, which is why they appear to be. And what you find is that for most animals in this list, you can in fact perform less than linear number of experiments, right? Which is very worrisome, right? So not only in the worst case are you stuck with this exponential barrier, but even what you can do is less than linear, right? So which is a very stringent constraint. Now you may say, well, you don't have to wait for the behavior to finish. You don't have to wait for a second. And here's this very nice paper from the 90s that showed that, for example, you can do fast visual processing. Even humans seem to be able to do it of the order of 150 milliseconds. So let's say we had neural proxies of behavior that manifested in 100 milliseconds, right? So I'm going to plot the average lifespan in units of 100 milliseconds. And that doesn't really help things too much. Things still stay around the linear regime, and for many it's still sublinear in terms of number of neurons. And again, already with many conservative assumptions thrown. All right. So I have maybe 3 or 4 more slides. So I'm close to being done. So I'm sorry, it's taking longer than the hour. All right. So to summarize, the results here suggest that in addition to current technological impediments, right, which are impediments of experimental technology, there exists another fundamental barrier, which is an algorithmic one to neural circuit understanding. Namely, this is the idea that we may not have efficient algorithms relative to the number of experiments that we can practically run on these individual animals. And going forward, therefore, the progress we make in understanding neural circuits is likely to be frequently driven by algorithmic constraints rather than limitations in experimental methods. Right? And one way of looking at it is also that you therefore need theory to sort of shrink the space of possibilities from your neural circuit. So there is a sort of one school of thought in neuroscience which says that you don't really need a whole lot of theory in order to figure out how these neural circuits are working. You just go in with your shiny new futuristic tools and you interrogate your neural circuit, which is what they call it, right? And then the answer pops out. But what they're saying is that without theory, you may not be able to shrink your search space sufficiently. All right, now what? So you could say, well, what are questions for which there are fast algorithms? That's one way. And on the other hand, you could say, well, even for these questions, are there subclasses of circuits that you can answer fast? Right? And I'm going to skip a bunch of these things. All right, and I just have possibly 1 or 2 more slides. So neuroscience. Again, so this is going back to neuroscience at cellular resolution has largely been correlational so far. But we need systematic theory to define causal notions of mechanistic circuit understanding. And a lot of this work, again, this is very active area of work. And there is a lot of conceptual progress that needs to be made, certainly in my opinion, right? And again, so now I'm sort of getting into the realm of speculation, which is going to be the last few slides, right? So and not just conceptual, but also one needs to throw in algorithmic tractability, especially in the neuroscience context. And one needs to see to what extent we can circumvent this algorithmic barrier. All right, now getting to sort of the more general speculation with respect to AI and so on. So I'm sure it wouldn't have escaped many of you that this sort of parallels the analogous question with contemporary deep learning systems, right? Wherein we have these systems that work very well, and yet all our old theories suggest that they shouldn't. All right, and we have new work that is beginning to try to show why they are working well, but even there, we are still barely scratching the surface of why it is that they are working and how should we understand them. So so what is the answer like? So with respect to, say, understanding deep networks, could something similar be true? And what I'm going to do is not answer it right away, but show you an analogy with Turing machines, right? So this is Rice's theorem from computability theory, for those of you who remember. So so this is sort of a generalization of basically the halting problem, right? So Rice's theorem says that. So let me sort of skip all of this and let me explain to you what Rice's theorem says. So suppose you had you have Turing machines, right? And now Turing machines have infinite memory and so on, but each Turing machine has a finite length description, right? So what you can then do is you can take that description of a Turing machine and feed it to another Turing machine, right? Which is a fascinating thing, right? And you can construct things such as universal Turing machines, which can simulate any other Turing machine. But what this allows you to do is to also try to build Turing machines, or at least think of building Turing machines that can answer questions about other Turing machines, whose descriptions you plug into them, right? And now, as you know, each Turing machine has associated with it a certain language, right? So now you can think of partitioning the set of languages into two, all right? The set of all languages, all right? And now you might want to build a Turing machine, which given, yeah, oops, sorry. I'm almost done. So hopefully some of you can stick around for Q&A. So the point I was trying to make is that in general, even telling which partition of these languages a given Turing machine falls in is undecidable. That's what Rice's theorem tells us. And so so it's possible that similar constraints exist, and therefore in a certain way, understanding several aspects of Turing machines are undecidable. So so my speculation is that something similar might be true of sufficiently complex AI systems. And this is again something that I haven't seen being shown in any rigorous way, and hopefully I thought this will stimulate some discussion. And also there is, so here was a recent paper, well, now 2 years old, a paper by Tim Lillicrap from DeepMind and Conrad Cording from Pennsylvania, UPenn, and the title of it was "What Does It Mean to Understand a Neural Network?" And here again they make the point that, look, with conventional deep nets, the code that trains them is, you can write it in a matter of pages. Yet the network itself is, has millions, possibly billions of parameters. And so here they argue that, look, even we should even abandon the goal of trying to understand the entire network, and instead just be content in understanding just the learning algorithm, as it were. All right, and which is sort of a very, very defeatist argument, but still it was interesting, and this is certainly a view that is increasingly being held by many in the field, both in neuroscience and contemporary deep learning. And so there's a take-home message. So non-existence of tractable algorithms for neural circuit interrogation could pose a barrier to comprehensively understanding how neural circuits in the brain cause behavior. And here is a preprint of this work as well. And I'm happy to take questions. Thank you.
Okay, any questions? It's quite well past time, Venkat, so people...
Yes, I know. I apologize. I think the background slides took more time than I had.
Yeah.
Yeah.
So I had a question.
Yeah.
So let's say that for once we let go of this, we're ready to do a trade-off between the computational limits and just implementing a fully functional neural circuit, right? Like it can be as big as possible, maybe not as compact as we would like it to be. But yes, it is practically possible, right? Like even as of now. Would you say that?
So are you talking about the sort of neuroscience case or the biology case or the
Computational case.
So
I want to build a computational model that is as functional as the human brain, right? You're saying that I probably cannot implement like the exact neural architecture in the human brain because of algorithmic.
No. So I'm saying that you cannot. So going into reverse engineer it is the problem.
Reverse engineer what?
Reverse engineer the network. So in other words, you might be able to train it and might work well, but if you want to go in and understand how exactly it's working, right, which is the question both in contemporary deep learning as well as in neuroscience fundamentally, right?
Right. Yes, I understand.
So that was so I mean training itself. I mean we train lots of deep learning models that work so well. In fact, these NLP models with billions of parameters, which depending on who you ask, work to varying levels of efficiency and so on. Accuracy.
So for a totally so for a person for us who actually just want to models that work, why is it so important to understand first of all like how does it work?
Right. So yes, okay. So there are several. This is a great question. All right. So there is this whole whole sub area of bias in AI.
Just a quick question.
Yeah.
I mean, within the question itself, I have a little doubt. Sorry, who was asking the question? Was it Mayur or?
It was Palash, me.
Palash. So Palash, when you said that for those of us who are interested in models that work, why is it important to understand it? How do you know that it works before you can understand it? I mean, in the sense that, how do you know that it works?
Sorry, in the sense I meant that I can probably all that matters is probably like if I have a functional model, a model that works, right?
No, but when you say it works, what do you mean? I mean, are you going to say
which has all the functional capabilities of the human brain, right? It like
no,
probably has all the...
So you mean it's like logically, in some sense, equivalent to the human brain?
Yeah. From a
but that we
purely
cannot.
functional
We won't be.
Right? It has the same capabilities as a human brain. Probably not the same. Like
No, but maybe are you talking in? I mean, the reason why I'm sort of quibbling over here is because I think these are easier statements to make than to actually follow through. So when you say functionally equivalent to the human brain on every kind of task or just on some specific tasks?
Let's say all sorts of tasks. Okay, let me take an example. Like, let's say there's a person. Let me draw a computational analogy. It takes inputs via its senses. It does some functions. It returns some outputs, right? So I'm just defining it totally functionally, right? There's no biological.
No, I understand that there's no biological aspect to it. I mean, if you're just talking algorithmically, so you're saying that these two functions are somehow the same, equivalent in some input output.
So the question is that so practically it is possible to build.
How are you going to? So I still have a difficulty in saying, I mean, with the human story, I mean, I don't know, that gets it even worse. But suppose you just say that you have a particular function that you're trying to approximate and or identify. Forget the human involved. Presumably the human, it doesn't matter to you so much. Since anyway you're not interested in the biology, that it's a biological organism. I mean, it can be just any function, isn't it?
Yeah. So the reason why I said this because Venkat sir mentioned about a mechanistic approach towards building what do you call these computational models, these systems. And so what that means is that let's say that I even forgo about the biological architecture, but I still know the functionality using psychology or purely data-driven approach, let's say.
Well, I mean, it's a bit difficult for me to still understand what exactly you mean that when you say that I know the functionality. If you know the functionality, why do you? Why can't you just?
I have a situation. Let's say if I somehow I have a situation, let's say, and I know that an entity, a human or anything, would react in this particular way. That's what I want and let's say I define this entire spectrum of functionality. That is what I meant.
I still don't want to bring humans into it because I don't know.
Okay.
When you are saying that a human will react in this particular way, unless you're pretty sure of that. I mean, I'm happy to bring humans into it, but until you're quite sure that all humans or some humans or one human or what, or your mom or what? I mean, whom are we talking about? So let's leave humans out of it. Let's leave any particular chimps out of it, whatever. But let's just say it's a function which has some inputs and some outputs, and you have some data which you so you're trying to understand or identify this function, and with some inputs and outputs. And when you say that you have a model that works, does that mean that given the same inputs, that model produces the same output?
Yes, exactly.
But that's not sufficient to say that the model worked unless you have some mathematical guarantees, isn't it?
So can I ask the same question? Like, what do you mean by work?
Well, why should I answer that? Because you use the word.
So what I mean by work is that
I mean, I have an idea of what I mean by work, but I'm not going to tell you.
The brain is a black box to me, and so is my computational model. But all I know is that it gave me the same output when I gave it the same input.
Yeah. So finite amount of input, or I mean, is the whole world finite? Your input set of inputs is finite. You know all the things, all the inputs, and you're
okay. So even if I build a system that is not as sophisticated in terms of its ability to take the entire perceptual inputs that a brain can take, any biological system can take.
Well, let's not worry about that. Let's just stay with this input and output of the function. I mean, let me just get past this point, and then I'll worry about the brain and eyes and all that sort of thing. So just the input and output. We have a black box. We don't know what's in that black box. It's a function. We're told at least that it's a function. We don't. In general, we may not even know that. It might be a relation. But let's say that it's a function. Then what happens? I mean, you're given some data, you see some output, you build a model, and you're going to say that your model works because for the data that you've seen, it has the same output. Is it? Is that your?
Yeah. And my point being that I can probably build a practical computational model that is as sophisticated as I need it to be, probably not as sophisticated as any brain. And I don't even need it to be as sophisticated as a human brain.
No, forget the brain. But even this thing where we're talking about just a function of some kind. So you're saying that based on the data that you've seen from the black box, which you're trying to identify, the model that you have does the same thing, and that's what you mean by works, yeah?
Yes.
Okay. Okay. Well, I think there's a lot of theory that tells you that that's there are to just say based on essentially what you're saying is if you are completely consistent with your training data or for your model builder, then you've identified the function.
I've okay, let's say I've not identified. I've approximated it, but it's sufficient for me.
Approximated it, but suppose you saw only 3 instances to your black box. You can approximate it. Is that good enough? I mean, when is it that you're going to say that you've approximated it?
When it's up to all my standards, and I can define those standards.
No, but you can, if you're going to keep moving the goalpost like that, when you say I can define the standards, what do you mean? I mean, it's so I told you, I've given you 3 instances to the black box. It came out with 1, 0, 0. You have a model which takes those 3 instances, returns 1, 0, 0. Have you identified it or not?
I don't know.
Is it working according to you or not?
I don't know.
You don't know. Okay, so then you don't know that it's working even with. So let me make it even simpler. What about with 1 instance? Would you be happy with it?
Yeah, I wouldn't be able to.
Yeah, okay. So what about with 1 million instances? You got it right. Then would it be okay?
Going by the logic, maybe no, yeah.
So difficult then to say that. So when you say that I for those of us who are interested in models that work, then if you can't define what work is in the first place, then what are we going to do now? I mean, now Venkat can answer some question, I suppose. Venkat, you can assume that Palash has some notion of works, and you can try to answer it, but I think it would be difficult because if you don't know what a working model means, then because you're probably trying to identify the model completely accurately. I mean, the model, when you say a model of the causal model, you're saying that it has to be the right model, yeah?
Palash, was that directed towards you? Or? So maybe let me try to. So my understanding of Palash's question was, look, why do okay? Let me paraphrase it, and Palash can say if that's what was the import of the question. So I have this, let's say, deep net model that works for object detection or object recognition, and so why should I worry about understanding it? Was that the question? Is that
yeah, okay, yes, maybe
in a way, right?
Yes.
Okay, so several answers. So one, so many of these sort of systems are being deployed in very sort of critical areas, so including things like medical applications, wherein your AI system has to make a certain inference about let's say a patient's prognosis and so on, or even in things like criminal justice, right? So you have these cameras that are all over the place, and they're trying to preemptively identify criminals. Of course, these are all of questionable questionable utility, in the sense of whether it is even in principle a good idea to do it or not is one question. But there there are people who are building these things, right? And so if and in fact, in many cases, it's even been shown, for example, that in the United States, such systems disproportionately pick out African Americans, right? And sort of flag them as potential criminals and so on. So there is this real issue of bias in AI, and so when an AI system is being used for something like criminal prosecution and so on, it is important that we understand how it made the inference that it made, right? So that is one type of answer, right? And so in many of these things, in order to deploy them in any meaningful setting, society requires that we are able to provide ironclad guarantees that the automated system that we have isn't just introducing another kind of bias, but one that we cannot really detect or easily tell, right? So that is one. Number two also is that in order to and there's this whole talk of AGI, right? Artificial general intelligence. In order to make progress towards that, it's important that we understand what the current systems are doing and what is lacking and how we might be able to get there. So I think towards that end also, getting to an understanding might help, right?
So is this to say that a proper understanding of the theoretical model or theory in general, a good understanding of theory in general is the solution to all these
so I don't understand. So when you say proper understanding of the theoretical model of what?
Of the brain.
Of the brain, okay. So
the reason I say this because now.
Okay. So wait. You were talking about AI systems, and now you're talking about the brain. So that's a different answer.
No, so yeah.
So wait, is it AI systems or brain? So brain, I can give you the answer, right? So one, of course,
the point is like building computational models of the brain.
Of the brain. Why is it important?
Would understanding the theory of the brain and then implementing into the computational systems is the solution?
No. Look. Hold on. So you are asking 3 or 4 different questions. So let me. So the first question was why should you care about understanding AI systems, which I think I gave my answer. Number 2 was why should we?
No, I actually come back to that either. So that's connected. These are all connected questions.
Okay, so let me try to, or if you want, finish your question if you like.
No, please go ahead.
Yeah. So with the brain, well, why should we understand? Well, one, because it's there, right? It's a basic science question, and even with all of the AI stuff we have, a 1-year-old can beat most any AI system on the planet. A 1-year-old dog can beat many AI systems on many tasks. And number 2, there's also the question of psychiatry. All right, so psychiatry has been a very sort of so many psychiatric ailments, all right, have very ad hoc both diagnosis criteria as well as treatments are fairly ad hoc, right? And there is a belief that in many of these cases these are caused by what people call network pathologies, right? So if we understand the working of these networks, then there is we are still not close to doing it, but the hope is that eventually we can get to a point where we understand the working of networks of neurons in the brain well enough that we can suggest a sort of credible treatments for many of these psychiatric illnesses. And so that's one other. Not I mean, everything doesn't have to be in the service of AI. Right? Neuroscience is not necessarily in the service of AI.
Yeah, I understand that. So neuroscience is one thing, but then why should my computational models be why should I care about the implementation of this exact neural circuit computationally?
Okay, so I think we are running out of time also. Palash, maybe you can sort of talk to Venkat separately. I think there are 2 confusions here. One is AI models for engineering and AI models for science. And I think Palash's questions, from what I can understand, are to do with AI models for engineering, like some object recognition task or some language sentence task or whatever, and a model for for scientific purposes to try and understand how the brain works and what how the human brain or any brain or hydra brain or whatever works. And for that, you have to build a model of that brain, or you have to be able to do experiments with that organism to be able to understand how that model works. So if you're going to build an in silico model completely, then it has to reflect, at least in my view, the underlying thing that you're trying to model. Just like if you're trying to model, I don't know, some kind of organ of any kind, you have to build a fairly sort of model which is reasonably high fidelity. But for the engineering point, you don't have to worry about the human brain as such. I mean, just like for building airplanes, you don't have to understand how a bird actually sort of flies, I suppose, as long as the airplane flies. But then there are different questions that then arise on the engineering front, that most engineering models are fairly well tested and fairly well proven on various sensitivity and calibration type performances. So you have to understand the engineering model to make sure that it doesn't go bananas on certain kinds of inputs. And you have to understand so in the second case, it seems to me, I mean, pointless to build a computational model to understand the brain without reflecting the underlying mechanisms of the brain. I mean, what is it that you're trying to build then? If you're trying to mimic the human brain on something, that's a different problem. But either you're trying to, so for the purely engineering task of, say, recognizing number plates, probably you don't need to understand how the brain recognizes number plates, as long as you have a machine that can do it. But you can make sure, you have to be pretty bloody sure that it's doing it. I mean, and it's not going to say suddenly that the number plate's a giraffe or something. Yeah. So there are two separate cases. Venkat's talk is about the AI for science, understanding of the brain as such. So anyway, we're a bit out of time. So is that Palash, could you follow up with Venkat if you want to?
Yeah,
Yes.
I'm happy to follow up.
If there's no other questions, I mean, I think we've already gone an hour and a half.
Yeah.
So, I mean,
Let me
very
stop the
interesting.
recording as well.
Obviously, the talk and the area is very interesting, but and that's why there would be, it takes a long time to for the talk as well. But I mean, I'm happy enough for it to go for 1.5 hours, but maybe not 2, I suppose.
Yeah,
so,
yeah.
So thank you, Venkat, for a very interesting talk. Yeah, so I'll just the rest who can drop off. I mean, there's no need to stay on. I suppose.
Let me stop the recording as well. Yeah.
Yeah.
So it doesn't.